Hello, my name is Laura Lewis. I am a sixth-year PhD candidate at Harvard University, and today I'll be talking with you about my dissertation research exploring social attention and long-term social memory in zoo-living bonobos and chimpanzees. So complex social life requires the ability to keep track of others, including pro-social group mates, aggressive competitors, potential mates, and also opportunities for social learning. This requires mechanisms for recognition, social attention, and long-term social memory. The first aim of my dissertation research is to investigate capacities for recognition, attention, and memory in our closest living phylogenetic relatives. And the second aim is to identify selective pressures and social experiences that may shape variation in these mechanisms. I study chimpanzees and bonobos in a comparative context because I think it's important in order to understand the selective pressures that may have shaped their social cognition in similar or different ways, and also our own, as we share a last common ancestor with them that lived between 5 to 7 million years ago. So I know we're all probably very familiar with these species, but just as a quick overview, first, chimpanzees are male dominant. So males make the big group decisions and form coalitions with each other. They experience fission-fusion social dynamics where they can live in groups of up to 200 individuals, but these split apart and come back together at different times of the day. They experience female dispersal, so females upon reaching pubertal age will leave their natal groups and go and join neighboring groups in order to avoid inbreeding. And they also often have very hostile interactions with neighboring groups that can even be lethal. Bonobos, on the other hand, are female co-dominant. So females form coalitions with each other and occupy the highest ranks of the dominance hierarchy, typically. They experience also fission-fusion social dynamics and female dispersal, like chimpanzees. Interestingly, they have very pro-social interactions, often with neighboring groups. So they can play, feed with, groom with individuals from neighboring groups, and these interactions can last even for weeks at a time. So I worked with several different populations for this research. The first are chimpanzees living at the Edinburgh Zoo in Scotland. I also worked with bonobos living at the Plankendael Zoo in Belgium. And then I had the immense opportunity to collaborate with Dr. Fumohiro Kano, where he ran my research at the Kumamoto Sanctuary with chimpanzees and bonobos. I used eye tracking methods for this research. So eye tracking uses an infrared camera in order to measure the eye position and eye movement of a subject. So we showed 8 subjects pictures on a screen, and then the eye tracker, which is situated directly below the screen, can calculate where the participant is looking on the screen at any given time. Throughout these studies, we used unique calibrations for each individual in order to ensure that we're recording as accurate gaze measures as possible. And we also used a juice dispenser for maintaining attention, which delivered a small amount of diluted juice in order to keep the apes' heads as still as possible. This is what the actual setup looks like. So most importantly here, this black box is the eye tracker, which is situated directly below the monitor screen on which we're showing trial images to the ape participant here, drinking out of this juice dispenser. So the first study explored social attention to in-group versus out-group members. And we had 2 driving questions here. First is, do bonobos and chimpanzees attend more to their group mates or to strangers? And secondly, which selective pressures have driven biases in social attention to in-group versus out-group members in these species? Social attention defined here is directed visual attention towards conspecifics. And this is important for learning socially, mating, and avoiding aggressive encounters. The swift and accurate identification of outgroup members is of evolutionary significance, especially for species like chimpanzees, where these decisions can even be lethal. Chimpanzees and bonobos are therefore the ideal models to explore the evolution of social attention because they live in large multi-female, multi-male groups with dominance hierarchies. Their unique relationships are related to fitness, and they have these fission-fusion grouping patterns where they might not see each other for a while. So we presented 8 subjects with 2 static images on either side of the screen. One was an image of a current groupmate, and the other was an image of a stranger of the same sex. We chose images of neutral faces so that there weren't any emotional expressions that may be biasing attention. And we showed these images for 3 seconds at a time. We showed apes about 36 trials each, and 29 apes participated in this study. So I'm excited to show you these results in this manuscript that was just published last June. So what you're looking at here on the y-axis is this differential looking score, which is a proportional score measuring out of the total time looking at the images, how long were they looking at the image of the familiar groupmate versus the unfamiliar individual. So a score of +1 means they spent all of the time looking at the image of their groupmate, and a score of -1 means they spent all of the time looking at the outgroup individual, the unfamiliar stranger. What we have here is data combined from both chimpanzees and bonobos. And on the x-axis are the sex of the avatars. So on this left panel is the dominant sex, males for chimpanzees and females for bonobos. And on the right panel is the subordinate sex, females for chimpanzees and males for bonobos. What you can see here is that when looking at images of the dominant sex, chimpanzees and bonobos look significantly longer at the image of their groupmate as compared to the stranger. So chimpanzees looked longer at male groupmates and bonobos looked longer at female groupmates. And this pattern was also significantly different from when looking at images of the subordinate sex, where they didn't differentiate as much between in-group and out-group members. Thus, it seems that apes generally look significantly longer at images of their groupmates of the dominant sex. However, we found some interesting differences when we looked at the different populations. So again, for bonobos, they looked longer at images of their female groupmates. Both Kumamoto bonobos and Plancandel bonobos looked longer at images of their female groupmates and didn't differentiate as much between male images. However, when looking at chimpanzees, we find this opposite pattern in Edinburgh chimpanzees. They looked longer at images of their male groupmates, but didn't differentiate as much between in-group and out-group females. However, Kumamoto chimpanzees spent more time looking at the image of the out-group males as compared to the in-group male. And this population is an interesting case because they only have 1 in-group male out of the 6 chimpanzees living in the group. And thus we think that because of this social environment in which there's only 1 male in the group, this may also affect their patterns of social attention, such that they're more interested at looking at out-group males rather than the in-group male. Thus, these results support our dominance-driven discrimination hypothesis, which suggests that social attention is shaped by sex-based dominance patterns, which is similar to what we see in other species. For example, juvenile vervet monkeys and rhesus macaques also selectively attend to and gaze follow high-ranking individuals as compared to low-ranking individuals. So it also seems that from the patterns from the different conspecific populations, that social attention is not only driven by these selective pressures of dominance patterns, but also by the social environment and the socialization in which these apes live. The second study explores long-term social memory in these species. And again, we have 2 driving questions here. The first is, for how long do bonobos and chimpanzees remember their previous groupmates? And secondly, do they remember some individuals more than others? Long-term social memory is defined here as memory for others that can last for years. And this reduces chances of aggression, promotes cooperation, and stabilizes long-term social relationships. It's adaptive, especially for species with fission-fusion social patterns. We know that bonobos can remember the vocalizations of their previous groupmates for up to 5.5 years. And as humans, we have a remarkable memory where we can remember faces for over 30 years. And our social relationships also have a very powerful effect on memory. So in free recall, we remember others in clusters based on social relationships and social roles. Thus, in this study, we aimed to explore long-term social memory in chimpanzees and bonobos. And we used the same paradigm as in the first study, where we showed 2 static images on either side of the screen, except this time, instead of a current groupmate and a stranger, we chose images of previous groupmates and a stranger of the same sex. So we showed apes about 36 trials each, and there were 27 apes that participated in this study. We also ran a zookeeper survey in order to determine the social relationships and dominant status of these previous group mates. And thus we could use these keeper responses to test the memory of relationship and memory of dominance hypotheses by comparing the relationships and dominant statuses of these previous group mates with social attention towards these individuals as measured using these eye tracking methods. So I'm excited to share these very new results with you. Here on this y-axis, instead of the proportional score, we're using just a raw difference score. So looking time towards a previous group mate minus looking time towards the unfamiliar stranger. So a score of 3 means they were only looking at the image of the previous group mate, and a score of -3 means they were only looking at the unfamiliar stranger. And what you can see here is that combined, bonobos and chimpanzees look significantly longer at images of their previous group mates. And this did not decline over time. So we had images of previous group mates that they hadn't seen in almost 10 years, and they looked equally long at these images as compared to group mates that they hadn't seen for a shorter amount of time. Excitingly, we had some trials in which a bonobo hadn't seen her sister for 27 years. And I'm showing you her data here, this yellow dot, which shows that she was much above the model averages when looking at these images of her previous group mate, her sister, that she hadn't seen in over 27 years. We also found that, that their social relationships had an effect on their memory. So again, on this y-axis is the raw difference score. And on the x-axis is the levels of positive interactions, with 1 being high levels of positive social interactions and 5 being low levels of positive social interactions. And what we found is that apes looked significantly longer at images of their previous group mates with whom they had higher rates of positive social interactions. Thus, it seems that chimpanzees and bonobos recognize familiar individuals, both current group mates and those they haven't seen in many years. Social attention in Pan is likely driven by both sex-based dominance patterns and the current social landscapes, as it is in other primate species. Long-term memory of faces likely lasts for at least a decade in Pan species, and there seems to be better memory for group mates with whom they had more positive social relationships. Thus, it seems that our last common ancestor was likely able to track conspecifics across distant space and time. And early hominins may have used these mechanisms as they engage in increasingly complex intergroup interactions across distant environments. With that, I'd like to say thank you very much for listening. Thank you to the International Primatological Society for hosting this conference. Thank you to Chris Crepenier and Professor Joseph Call for overseeing this research and my collaborators. And thank you so much to the zookeepers and staff that were fundamental to this research and really supported me all along the way. Thank you very much.